Hello, this is Dr. Moore, and this video is about bacteria and viruses. In our day-to-day lives, I think we tend to associate bacteria and viruses together because they're both tiny things that make us sick. But from a biological perspective, they're really quite distinct. Bacteria, along with archaea, are prokaryotes. That means that they are single-celled organisms and that their cells lack membrane-bound organelles like a nucleus or an endoplasmic reticulum. They, along with eukaryotes, are definitely living things, whereas viruses, most biologists don't consider to be living things. They don't have cells, and they require something that does have a cell, a host, in order to make copies of themselves. But there's no doubt that viruses are biological. So if bacteria and viruses are not that similar, why are we talking about them in the same video? And for that matter, why are we talking about them in the biotechnology unit? We're talking about bacteria and viruses today because they both happen to have really interesting quirks in their biology. And if we understand those quirks, we can use them as tools to make the biotechnology that we want. So this video is going to go through 7 things that you need to know about bacteria and 5 things that you need to know about viruses. And we'll start with the bacteria. First thing to know is that bacteria are really small. I'm a big fan of this simulation from the University of Utah, which shows you just how small the bacteria are. So you can see we're starting with familiar household objects like coffee beans and grains of rice, and we're going to zoom in. So we're going past a grain of salt. Here we've arrived at the first eukaryotic cells. So the amoeba, the paramecium, the human egg, these are all eukaryotic cells. They have a nucleus and other membrane-bound organelles. Let's keep on zooming in. We're getting to some smaller eukaryotic cells like a photoreceptor cell, a skin cell, a red blood cell, a sperm cell. Yeast is still a eukaryotic cell that's a fungus. It's only when we're down to the size of the mitochondrion that our first bacteria shows up. And the fact that bacteria and mitochondria are about the same size and shape is more evidence in support of the endosymbiotic theory that our mitochondria and chloroplasts in plants evolved from prokaryotes that came to live inside of larger prokaryotic cells. So bacteria are pretty small. The next thing that you need to know about bacteria is that they don't have membrane-bound organelles. No nucleus, no lysosome, no endoplasmic reticulum, no Golgi apparatus. What they do have, however, is a cell wall, and just like the cell walls of plants and fungi, the cell wall of bacteria is made out of complex carbohydrate polymers. They have a cell membrane, they have cytoplasm, and they have ribosomes. They also have circular chromosomes. So when you think about the chromosomes in a human cell, we say you have 46 chromosomes. Those are 46 linear chromosomes, or 46 individual strings of DNA that each have 2 ends. Whereas bacteria have circular chromosomes. So their chromosomes form a ring. And most bacteria just have one main chromosome in their body. You can also see in this diagram that prokaryotes can do simultaneous transcription and translation. So there's no reason that they can't be making an RNA copy of their DNA at the same time that mRNA is getting read by a bunch of ribosomes, because there's no nucleus, so there's no physical separation of those 2 processes. Whereas in eukaryotes, transcription occurs within the nucleus and translation occurs outside. Another thing you can see in this diagram is that there is no mRNA processing. In eukaryotes, our mRNA has to have a 5' cap added, a methyl tail or poly A tail added rather to its 3' ends, and we have to remove the introns, so those non-coding regions of the mRNA transcript before it gets sent to the ribosomes. None of that is occurring in a prokaryote. Right after transcription, the mRNA is ready to be translated. Bacteria also have plasmids, which are pretty nifty little things. A plasmid is just a little ring of DNA, probably contains only a handful of genes. It's not the big main circular chromosome that contains most of the genes that the bacteria needs. It's just a little thing with a couple of extra genes. And those plasmids will play an important role in horizontal gene transfer. Horizontal gene transfer refers to the ability of bacteria to give and receive genetic material in their lifetimes. It differs from vertical gene transfer, which is the gene transfer that's associated when parents give their genes to their offspring and their offspring to their offspring. Horizontal gene transfer is getting new genes, but not from your parents. There are 3 ways that bacteria can do this. The first one is called transformation. So bacteria can pick up DNA that they encounter in the environment. This would be like if you could walk down the sidewalk and find a piece of DNA and just stick it into your body and see what happens. The second one is transduction. This is when viruses carry DNA from one bacteria to another. When we talk about viruses in a minute, you'll see one of the things they do is package some genetic material within them and then inject that genetic material into another cell. And so if a bacterial gene gets mixed in to the virus, that gene can travel to another bacteria. And lastly, there is conjugation. This is when 2 bacterial cells come together and one bacteria will give its plasmid or a copy of its plasmid to another bacteria, which will pass along a few genes that were on the plasmid. This would be like if when we had sex, we got new genes from our partners that got incorporated into our bodies. So that's what you need to know about bacteria. Let's think now about viruses. What you need to know about viruses, the first thing is that they are really, really, really small. So let's go back to the University of Utah size and scale demo here. So we had finished here where we were looking at a bacteria and a mitochondrion. We need to keep on zooming in to see viruses. Here you can see some viruses and they're hundreds of times smaller than a bacteria. We got the measles virus, influenza virus. This one is an HIV virus. We can keep on zooming in. This is a rhinovirus. This is the kind of virus that causes the common cold. It's about the size of a ribosome, really, 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 really tiny. And the reason that they are so tiny is that there's not really much to a virus. It's just a little bit of genetic material that could either be DNA or RNA. And then that genetic material is wrapped up in a coat of proteins. And sometimes the virus will take a little bit of the cell membrane of the cell that it infected. But that's it. That's all they've got. That's not enough machinery in order to make a copy of that entity. And so viruses must use a host cell in order to replicate. This diagram showing you a general description of the process, where the virus will mount itself on the outside of a host cell and inject its genetic material into the host cell. The host cell then uses its normal cellular machinery to read that genetic material and make the proteins that result from it. And lo and behold, those proteins are going to assemble to form more viruses, which eventually rupture out of the cell and go look for more host cells to infect. There are viruses that attack bacteria. These are called bacteriophages. So the image that I've been using in this talk shows an E. coli bacteria, shown in goldenrod, being attacked by multiple green bacteriophages, which are the viruses that are attacking the bacteria. And lastly, some viruses use RNA instead of DNA as their genetic material. These viruses are called retroviruses because they work backwards from normal living organisms, which usually use DNA to code RNA to code for proteins. But in a retrovirus, the RNA is read backwards by a really important enzyme called reverse transcriptase because it does the reverse of transcription. And that reverse transcriptase will take RNA and make a DNA molecule using the RNA as a template. And then that DNA can be transcribed and translated into proteins via the normal methods. So I'm going to end by highlighting 3 ways that viruses can be used in biotech. The first one is gene therapy. So what viruses do is inject genetic material into cells. That means we can use viruses when we have genetic material that we want to inject into a cell. So if somebody has a disease because they don't have a functioning copy of a gene, we can use a virus as the vector to get that gene into the cell of the afflicted person. We can also use viruses as a treatment for bacterial diseases. This is called phage therapy, when we use bacteriophages to target specific disease-causing bacteria. And this is a better approach, if we can get it to work, than antibiotic treatments, because antibiotics will kill all bacteria, including the mutualists and commensalists that are either beneficial for us or don't really affect us at all. Whereas phage therapy would affect just the target bacteria. So we could kill just the bacteria that are making us sick. And a third use for viruses in biotech is that nifty enzyme reverse transcriptase, which comes from the retroviruses. This allows us to make DNA from RNA. And why would we want to do that? Well, one reason we would want to do that is that we can make DNA that doesn't have introns, because the mRNA transcript has had its introns removed. And so we can use reverse transcriptase to make a DNA transcript that doesn't include the introns. And this would be useful to us if we wanted to do something like put the insulin gene in a bacteria. Because bacteria don't have the ability to remove introns, we need to give them a copy of the gene that has the introns removed. And reverse transcriptase is one way that we can accomplish that. So this was an introduction to the biology of bacteria and viruses, and you'll get to see over the next week or two how their biology allows us to use them as tools.